welcome to another episode of the Incubator at the Bench. I'm really excited for our interview today with Dr. Jackie Lajiness. My name is Ben Fensterheim. I'm an instructor in neonatology at Children's Hospital of Philadelphia. And I'm going to pass it over to my co host, David.
Thank you, Ben. My name's David McCulley. I'm a neonatologist and a physician scientist at the University of California in San Diego. I study developmental biology of lung and pulmonary vascular development. And yes, I'm also very excited to be co hosting this show today and to be talking with our guest, Jackie Lajness from Indiana University. I met Jackie through the Midwest Society for Pediatric Research, and it sounds like she is helping to host that meeting that's coming up in Indianapolis in the next month. And so we're really excited to talk with you today, Jackie, and just to hear about your work and ask you some questions just about how your career got started and things like that. But just so that our audience knows a little bit more about you, would you mind just saying who you are and like what career stage you're in and just like a brief introduction into the type of work you do?
Of course. Well, first of all, thank you, David and Ben, for having me on. I'm so excited to be here. But I'm a neonatologist and a physician scientist at Indiana University. I started my lab right after fellowship. So 3 years ago. So now I'm an assistant professor, and my lab looks at how the maternal immune system can contribute to preterm birth and how that affects the in utero environment and what effect that has on the development of the neonatal immune system.
Awesome. That's perfect. Thanks, Jackie. Just to begin with a little bit, can you talk about what motivated you to study that line of research? It's really interesting, something that we all wonder about, like what causes preterm birth, but it's exciting to think about it in the way that you've been thinking about it and to use basic research to try to understand it.
Sure. Yeah. So I was kind of surprised at how much there still is to learn about what causes preterm birth. And of course, no one thinks it's one mechanism, right? There's lots of things that lead to preterm birth. But I was particularly interested in this subset of moms who are otherwise healthy, right? There's no huge, overwhelming infection. They don't have horrible preeclampsia that puts mom and baby at risk. But they're still going into preterm birth. And so my thought was, well, if we understood a little bit more about the mechanisms and potentially how mom's immune system is leading to this kind of inflammatory environment that's starting the kind of process of labor and delivery, if we understood that better, could we, one, identify those moms and two, intervene in a way that would be helpful for both mom and baby and improve outcomes? So that's kind of where I got started.
I think that's really fascinating, especially because it sounds like not only does what happens before birth extend to birth itself, to delivery, but also it sounds like to life after birth, to neonatal life, maybe life in the NICU or maybe life at home later. Um, how have you been able to study such a broad topic? Like where do you find the areas of focus are in studying immune development through from maternal to neonatal life?
Yeah, it is a pretty broad topic. So right now I work on preclinical models. So I have a mouse model where we deplete T regulatory cells because we know that in humans, moms who deliver preterm without other complication tend to have fewer Tregs, and they tend to have Tregs that don't function as well. So it's kind of this model of a loss of tolerance. So I took that model and kind of isolated that in a mouse model. So I deplete Tregs in the third week of a mouse pregnancy, because if you do it earlier, you just lose all the babies. And in working with that model, I found that yes, they deliver preterm and that there are definitely changes to the mom's immune system. One of the things I look at is kind of cytokine signaling, and IL-9 is something that I've become interested in. But in addition to the changes that are undergoing in mom, I think that we're starting as a research community to understand and become more interested in how the fetus interacts with the in utero environment as well. And so I also look at the what changes are happening in the fetal liver during this time, because some of those cytokines cross the placenta, or there's other signaling factors that go from mom to baby and can influence the development. So, like my fellowship work was looking at how neonatal dendritic cells change when the mom has allergy. And so just being in that in utero environment sets them up and kind of primes those dendritic cells for changes postnatally and predisposes them to developing allergy. So I started looking at same kind of thought process in this model of preterm birth. And I find that there are definitely some changes in gene expression, as well as kind of the profile of the immune cells in the fetal liver that I can see as early as embryonic day 16 and a half. And so my next kind of focus will be on what that actually means for those pups that are born early and have those changes and what functional relevance that has.
That's great, Jackie, and has huge implications. Um, Obviously, very broad still, but I mean, amazing that you're able to do this work and understand these mechanisms in more detail. Before we get really deep into it, because I think we both have a lot of questions about this, can we just talk about your like kind of background and training a little bit more and like how you got to the position that you're in now? Just like where did you learn about the possibility of doing physician scientist work? I know you have a PhD. How did you pursue that path? Just could you talk about that a little bit?
Of course. So I first started doing research in undergrad. So I came in as a self-appointed pre-med student at Hope College, which is a pretty small liberal arts school in Michigan. But pretty soon after starting school, people were like, "Oh, you know what? Doing research really looks good on your med school applications." I was like,
Mm.
"I can do that." So, as a freshman, I went to the
Yeah.
handy dandy website on Hope researchers, and I found that one of my chemistry teachers, Dr. Krieger, he had a lab that looks at laser spectroscopy. That's what he works on.
Wow.
And so I reached out to him and I started working in his lab, and I was with a bunch of other really phenomenal students, right? We're all undergrads. And no one knows much about laser spectroscopy as an undergrad. So it didn't really matter. I wasn't at a disadvantage. So I lucked out in that Brent was a phenomenal teacher. He really loved building lasers, teaching, and I really enjoyed, learning how to build a laser. That was cool. But the intricacies of experimental design and being able to control the variables that you can control and understand what you can't control. And just, I fell in love with the freedom of being able to ask questions and actually go and find out the answer and design the experiment that'll allow you to do that. So I really enjoyed my time. And not only did I get to work with great people, but I also got to present my research at conferences. So I went to a few national conferences. I applied for a grant, which was great. And fortuitously, I ended up on the way to a conference sitting next to a professor from Case Western. And she clocked me pretty soon after I got on the plane because, of course, I was, carrying that telltale
Poster tube.
poster tube. Exactly. You know exactly what I'm talking about. And so we started chatting. She was asking me about my research and my career goals. And I was telling her I wanted to go to med school. And she's like, well, I mean, why can't you do both? And I can remember like thinking in my head, wow, that sounds really intimidating. How could you possibly be good at both? And realizing that I couldn't say that out loud. But that's kind of what I recognize as the time that planted the seed for the idea of a physician scientist career path.
Wow, that's amazing. I mean, it's that interaction with your mentor as an undergrad sounds really important just because that observation of just, getting to see somebody who's developed this career doing research, asking questions, designing an experiment to answer that question, test a hypothesis, and control variables so that the conclusions, seem like they're justified. But then also just kind of a little bit the chance of, you got to meet that person sort of just because out of a little bit of luck. And then that interaction on your trip also sounds like is just, a little bit by chance. But those things can be so meaningful. And I think, if you just connect with people at a really early stage sometimes, even though it may not feel that influential, it can have a really big impact on the person's thinking and like just opening doors and what they would consider for the future. So then I take it you learned a little bit more about like MD PhD path and then applied in on that route, or how did that go?
Yeah. So I had some friends who had at this point already gone to medical school, and we would have conversations about like, how's it going? And they would talk about how they really like taking care of patients. But at some point, you run up to questions that there is no answer to, and you really don't have much of a recourse as someone who's just in clinical care to be able to address those or improve kind of the future quality of life for your patients. And so that was something that really meant a lot to me. And so I started thinking more seriously about the very long career trajectory that is the MD PhD. But ultimately, when I applied to medical schools, I applied only to combined degree programs. And I met a lot of really wonderful people just through the interview process and kind of saw how people who have started a career as physician scientists, how they make that work. Wade Clapp was the director of the MD PhD program at Indiana at the time. And he's a tremendous example for me personally of what it means to be a physician scientist. And he was one of the most genuine people and so excited about science and willing to mentor people. And he was a big part of how I ended up at Indiana and why I've stayed here for kind of subsequent kind of career development. And so, yeah, a lot of these interactions sometimes come down to chance, but you take those opportunities and you run with it.
Yeah, one of the things I love about this story is it sounds like you approached the career from almost a conceptual love. Like you love the idea of discovery and you loved the idea of solving problems and doing experiments, collecting data, and setting up controls, or, rather than which some folks might approach the career as I really want to discover the answer to this specific problem as an undergraduate because maybe my mentor was working on that problem. I eventually found it interesting, and I think it's really interesting and fantastic to hear because I think that is a signal that you really will love the path. If you just love answering questions and doing experiments, and I think that is always going to be there. In the career, so I love that part of the story. So I'm curious then, when you transitioned to, and this is maybe thinking my own experiences as well, when you transitioned to finally becoming an MD PhD student, how did you find that transition from your undergraduate physics lab to medical school and graduate school at the same time? Was it surprising to you or what you expected?
I'm not sure I had a lot of preconceived notions about that because I had never worked with cell culture or animal models. And so I kind of approached it as I'm going to be learning a lot of new things, and there's definitely a steep learning curve. But as I kind of started medical school, I very much gravitated towards embryology and the idea of I like developmental biology. And so that kind of became my focus for the different labs I was looking in. And I ended up in a cardiac development lab. And I really enjoyed the science. I liked learning about we're in working with transgenic mouse models. I'm not going to lie, the we did timed matings, right? Because we did embryonic development. The first time they said, "Oh, just go check that mouse for plugs," I was like, "Excuse me? Uh, what do you mean?"
What exactly does that mean?
So it's definitely a steep learning curve, but those are things I still use in my research today. I work with transgenic mouse models. I do timed matings. Um, I like the idea of kind of the developmental aspect, which has been a continuing theme throughout my work. And so I took those experiences. And then as I kind of became a little bit more refined in my clinical interests, I knew I wanted to move more towards how maternal health affected the health of the baby and how the time of pregnancy can kind of be a really important kind of critical window for how some of these, changes are happening. So when I was in my fellowship, I was able to identify Joan Cook Mills as a research mentor. And she had this model of maternal allergy and, neonatal allergic predisposition. And we worked together to form a project where I could learn a lot of things about immunology and specifically the innate immune system. But I learned skills like flow cytometry that I had never used before. But I knew that that was the direction I wanted to take my lab. So I really learned a lot from that experience. And now I do immunology research. I'm still working on identifying as an immunologist. I still feel like I have a lot to learn. But Mark Kaplan is one of my mentors, and he's a just a great T cell biologist. So I'm still learning a lot. But now I use, spectral flow cytometry. So I feel like I've come full circle. I mean, now I'm thinking about, admission and emission spectra again. And it's
Oh,
like
wow.
full circle.
That's perfect. Can we talk just briefly about your timeline? Just I want to understand that a little bit. And I think one of the issues these days is like how to keep people who are on this track, interested and motivated and engaged realistically, given the challenges of clinical training. So, like, when you finished your MD, PhD, and then did residency, did you have much opportunity to like stay in a lab or like keep doing research, or was it something that you felt like you basically had to kind of put on hold, knowing that you would get back to it, hopefully as a fellow?
That's a great question. So for me, I knew I kind of wanted to take a little bit of a different approach for my fellowship work. So I approached residency. I did not do an accelerated or an integrated research pathway. Part of the reason was I had my first child in medical school. And so as a resident, I had a infant at home and I knew that I wanted to leave space for all of the things I would learn clinically in residency. And so I took kind of a month at a time opportunities where I would do research and I would go into a lab and I would focus on a technique. So I, learned kind of like, uh, I went and I learned CRISPR and I did kind of a short experiment where I worked in Laura Hainline's lab and I, did did some work with CRISPR just to learn that as a technique and learned some, in vitro studies, those types of things that I hadn't really done a lot of in graduate school. So I used that as a time to kind of identify techniques or areas of weakness that I would like to ultimately incorporate. And I used that time in that way. And then I applied for the PSDP program, so the Pediatric Scientist Development Program, which supported my fellowship research and actually gave me 2 years of protected time at the end of fellowship, which I thought was critical for transitioning to having my own lab right out of fellowship. And so that was kind of the trade off and the sequence that I did. So I came out of fellowship. I started as a tenure track. Assistant professor and started on a K-12 and then have worked towards kind of establishing my own research program, generating preliminary data, and applying for a K-08. And that's kind of been the trajectory I've taken in the first few years since fellowship.
That's perfect, Jackie. I think that's really helpful for people to hear. I think a lot of people find it challenging to try to use any time during residency to keep working on their research skills and development. And it does feel like it's fragmented, but I like the idea that you were like seeking out specific opportunities to kind of learn a specific technique. That That's perfect. And also, I think just talking about the PSDP and how much that can help people have a really solid mentor interaction, and also just like a sense of support that this is like a career path that you're going to be able to keep making progress on as you transition from fellowship and then into early faculty years. Um, I think it's great. Can we start talking more about your science now? I mean, I think Ben and I probably both have a lot of questions just about that. What questions are you really focused on right now, Jackie?
So right now I am focusing on in the bottle of Treg depletion during pregnancy. So if the Tregs aren't acting appropriately the way they should in pregnancy, what are some of the changes that are now predisposing to preterm birth? And so that's probably what my K award is focused on. And then kind of something that has grown out of that that I kind of see as a future direction that I'm going to start to grow is some of those changes that happen in the neonatal immune system developing in that environment.
I would struggle to be an immunologist myself, to be considered an immunologist, but I'll slowly try and pretend. Um, Treg dysfunction, it sounds like something that you are inducing in some way in the mom. Is it, or the dam? Is that right?
Yes. Yeah.
Yeah.
Right on all counts. Yes.
Um, so what type of exposures during pregnancy might cause Treg dysfunction in the way that you're inducing it in mice?
So the mouse model is a little bit artificial because we use a model that's a FOXP3 DTR. So these mice have basically a suicide receptor on all of their Tregs that I can induce depletion by getting them diphtheria toxin during pregnancy. Now, mice do not have diphtheria toxin on any of their other cells, so it doesn't cause problems in the rest of the mom. It just selectively depletes the Tregs. And so it's a little artificial, right? Because that's not what happens in human pregnancy. Typically, what we see is that there's a pretty big expansion of regulatory T cells during pregnancy, and the idea is that those cells serve to kind of dampen the immune response so that the mom can tolerate this allogeneic fetus that's growing inside of her, right? And so we don't really know if it's a that initial response isn't as high in moms who will go on to deliver preterm, but people have shown that at least at the time that they start showing symptoms, it's predictive of how soon a mom will deliver. If you look at the amount of kind of circulating regulatory T cells in her blood. So, the moms that have lower numbers of regulatory T cells, they're more likely to deliver within the next 48 hours. And there have been a couple studies that have shown that. And then if you look at the time of birth and you look in the decidua of those placentas that are a product of that pregnancy, you find that if you isolate the regulatory T cells from the decidua, they are not as good at dampening, T effector cell responses in placentas that were delivered preterm compared to even those that deliver at term and have already been through the labor and delivery process. So it is a little bit artificial, but it allows us to evaluate in a way that we can't do in human pregnancies to be like, okay, I'm getting rid of the Tregs. What's happening 2 days after that happens? What's happening, 3 days after that? So before birth is occurring, before that labor process starts, what are some of the changes that ultimately lead to that process? So there's no perfect animal model, but I like this model because one, it is immune mediated preterm birth. So we're identifying a kind of a pathology that if we could intervene upon it in humans would be helpful, right? So there are times when the baby just needs to be delivered for mom's health or baby's health. But in this instance, if we can stop the kind of loss of tolerance or kind of somewhere close along that pathway, can we actually prolong the gestation and keep that baby in a healthy in utero environment for longer?
Yeah, I mean, I think it's fantastic because inflammation has been associated with preterm birth in many different contexts, and this is to focus in on one of the most important cells in regulating inflammation, and how they change, observationally change, and then change in an experimental model to induce um, something like preterm birth. And I think it's a fantastic way to explore the role of these cells and general inflammation, whether some just generalized inflammatory event can precipitate preterm birth. And I completely agree with you about the utility of the studying the immune system just in a practical sense, because there are so many targets to to try and address what could be going on. Um, so, yeah, I think it's really fascinating stuff. I'm also curious to hear more about you mentioned that right around the time of birth, these cells almost like change their function, change their maybe location or maybe their phenotype. So they decrease in the blood and change their function in the placenta and the uterus. Um, is what do you feel like, or what is in your experience have you found that might cause these cells to change during pregnancy? And I'm kind of asking that from a question of, I know that there are many parts of the immune system that change during pregnancy because of pregnancy, and I'm wondering, do Tregs or does other inflammatory mediators change just because of pregnancy or just because of delivery even?
So, great question. And there are a lot of people that are still working on really defining the dynamic nature of the immune system throughout gestation. But yes, so Tregs, we know that they increase basically right after conception. And then we know that they start to tail off before the natural onset of labor and delivery. But we don't really understand the mechanisms of how that happens in humans or why. So, what signal are they responding to? Is it a maternal signal? Is it a fetal signal? Is it a combination of an interaction between the maternal and the fetal immune system? I mean, there's still a lot of things that we actually don't know about what starts labor and delivery process in humans, which is a very important thing. I mean, especially as neonatologists, we really care why this is happening.
I was wondering, just very simply, because we'll have a kind of a pretty broad audience. So in your model, do the mice actually deliver early?
Yes. So the mice deliver early, not all of them. So it shortens the gestational duration. And then if you arbitrarily define kind of preterm birth as embryonic day 18.5, only about like 20% of them deliver before that time. But if you graph it out, it definitely shifts everything. So they're delivering about a day to a day and a half before a normal pregnancy would deliver.
And it sounds like you're studying both the pups and the placenta, is it right? Is that right?
Yes, most of my stuff has been kind of focused the placenta as well as the fetal liver. So yeah, the pup response as well.
Does the placenta then, I mean, because basically you've lost or you're reducing immunotolerance. So, like, what do you see in the placenta? I mean, do you see like a evidence of inflammatory reaction in the placenta or some sort of like um, because of that loss of tolerance that the placenta basically is not growing well or is it dying early or what do you find?
The placentas aren't smaller or anything like that. I'm still working on through some histology, but people who have published this model in the past don't really show huge changes in like the placental structure. So you're
Okay.
definitely not damaging the placenta in a super obvious way. Nardi Gomez Lopez has even done some kind of ultrasound evaluation and showed that the blood flow through the placenta is intact, and the pups themselves are the same weight. So I don't see a really big IGR kind of phenotype. So overall, it seems like a fairly healthy pregnancy. I've looked at kind of the expression of certain genes that are involved in inflammation. So I mentioned IL-9 signaling has been something that I found has been interesting. inflammasome. So the NLRP3 inflammasome, which has been studied a lot in preterm birth work, is actually can be downstream of IL-9 signaling, and it is also increased in the placentas and the fetal liver of these mice. And so there are definitely some gene targets. I'm working towards kind of doing some more single cell kind of focused, so I can see which cell lines are contributing which changes and kind of understand the mechanism a little bit better that way. But that's kind of the direction I'm moving.
I'm curious in these in the pups that are born, even though they seem to their normal weight, and it sounds like the only thing that's really changed observationally about the pregnancy is the time of delivery in this model. Do you see changes in the pups later with their immune system or their growth and development after birth?
So growth and development seems to be fairly normal up until I keep them through weaning. So up until they're 3 to 4 weeks old, I follow them. Their growth trajectory seems normal. I'm working on kind of doing time points to actually look at their immune composition postnatally. Most of what I've done has been right around the time of birth or during gestation so far, but I do see changes that I want to follow longitudinally. But that's to be done.
Amazing. Well, I'm biased in my interest of, of that as someone who studies preterm infant immune development. Um, but okay, fascinating. Yeah, sorry. I think I jumped in on you, David.
No,
Sorry
not
about
at all.
that.
I was trying to keep it in like the clinical realm a little bit just because a lot of times, like, if a woman is coming in and threatening preterm labor, like one of the first things that we recommend as neonatologists is to give betamethasone. And it seems like in addition to helping mature lung function, it also acts to like decrease inflammation. Have you done that as well? Like you deplete the T regulatory cells in the mom and then just given steroids and see how that affects the risk of delivering prematurely and the inflammatory reaction? Or are you thinking along those lines or what do you think?
I mean, that's a great question. I haven't done that in our model yet, but it's something that we think about, right? So, there's so much of caring about inflammation that plays into how we take care of preterm babies and how we think about their comorbidities, right? Uh, If you're talking about neck or BPD, we know that that at least how that infant experiences inflammation definitely changes their course of the disease and their severity of the disease, right? It's a really big modifying factor. You can have babies that are exposed to what we think of as kind of the same stimuli, but have very different courses and different outcomes. And that's one of the fascinating things that we see as clinicians, right? And it's a really hard part about taking care of babies in the NICU sometimes, right? Is we can't always predict. But if we can understand a little bit more about, well, is this baby's immune system primed to react in a certain way based on their in utero environment? Can we be a little bit better about predicting, or can we tailor our treatments a little bit more postnatally to try and help them through those milestones that they have to face? And that would kind of be the gold standard for what I would like to contribute. I think that if we can understand a little bit better and how we approach and take care of our babies, we might be able to improve their outcomes.
That's exactly the question I was wondering about. I mean, I remember talking about that with Ben when we were talking about his work, where like we often talk about steroids in the NICU. We talk about it, for preterm delivery, but we also talk about it in preterm infants who have severe lung disease. And, the whole idea is to try to decrease the inflammatory reaction, but it does it through so many different mechanisms that we don't completely understand. And so trying to understand the actual mechanism of how depleted immunotolerance would lead to preterm birth might implicate a specific mechanism, and steroids might be effective, but maybe there's something that's much more specific that might not have the potential negative consequences of giving, um, like betamethasone.
Well, I wasn't going to ask another question. I was just going to continue your comment of your idea that is so good. I mean, there's so much anytime you see an anti-inflammatory antibody or very specific molecule be used, you can bet that in that disease they tried steroids first. And the targeted therapy was better. I mean, like, almost across the board in rheumatologic disease, there's examples from oncology, GI, like, steroids are often thought of as the first thing to do when you've identified an inflammatory event occurring. But in so many cases, getting specific with the anti-inflammatory intervention improves the therapeutic effect of what you're doing. And that's why I love how you're really anchoring on Tregs as cells, as mechanistic cells here, because they can be targeted specifically, and the cytokines that they make can be targeted as well. And I think it does raise a question, like, maybe we should be giving not just betamethasone for the lungs, but also an anti-inflammatory to try and slow the preterm birth down. Um,
Not to get too much into the weeds, but it's really interesting to start to think about how the context really matters, right? So you can have a single cytokine and it's never acting in isolation, right? But it can actually change like the kind of immune environment, right? So cytokines can change your expression of your receptors. They can also change expression levels of transcription factors, which then mediate those effects, right? And so you can have, a stimulus like LPS, right? We know that LPS exists in low levels, even in healthy pregnancies, right? But if you are changing the immune environment to make it more sensitive to detecting and responding to LPS, are you changing the, the tolerance level in that pregnancy for, what that small stimulus would, do? Does that mean that in a mom who has an inflammatory kind of milieu, that if she has that low level of LPS, that it's going to tip her over the edge and lead towards preterm labor and delivery, just because of the immune context. Whereas another mom could tolerate that same level of LPS and have no effect from it. So I think it gets really interesting, and that it's why I'm excited about the mechanism, is because the more we understand about the context, I think the more targeted we can be with our approaches. Like you were saying, Ben, like, is there a better target to be hitting that gives us the benefits without some of the side effects?
Is IL-9 something that you're thinking about? Is that, like, one of the things that you think if you were to block that, you might be able to rescue the risk of preterm delivery, or what were you thinking about that?
So I think it's a very interesting target because it's kind of been shown to kind of lead to preterm delivery. It signals through STAT3. So those are kind of like the mechanisms that I found is like IL-9 and STAT3, because STAT3 also signals through IL-6, and it also signals through, um, TLR receptors that respond to LPS. So that's kind of the signaling pathway that I'm focused on right now. But I think it's important to keep in mind that all of this exists in a context of a bunch of other cell types. And I think that not losing sight of that is important. And so that's kind of how I'm trying to focus my research, is learn the mechanism, but always keep in mind that this exists in a larger context.
I'm wondering that because you're talking about specific cells and cytokines associated with preterm labor and delivery, but there are other maternal reasons for needing delivery, like preeclampsia or infection. Do you find that there are different immune factors that are associated, or maybe if you found this or if others have found that there are different immune factors that are more associated with things like preeclampsia compared to something like preterm labor?
I think that's a really important thing to keep in mind. And yes, they are probably very disparate mechanisms. And the reason why I've kind of focused on like the immune mediated preterm birth is because it's if we can intervene in that, it doesn't hurt the mom or the baby. Because like preeclampsia, the mom is having a life-threatening reaction that if the baby isn't born, it's going to cause problems for mom and baby, right? That placenta is not healthy at that point. And so while those are really great things to understand and know about the mechanism, preventing that preterm birth might not actually be the best thing for that mom and that baby in that context. And so that's why I'm trying to focus on the population of moms that I think could benefit the most by actually stopping that progress without having negative consequences for mom or the baby.
Because you were thinking along those lines, so then to identify like a pregnancy that would be at risk, are you thinking of like looking or like somehow quantifying the regulatory T cells earlier in pregnancy or even in women before they were pregnant? Or what were you thinking about that? How would you identify like a pregnancy at risk?
So I would love to be able to appreciate the dynamic nature of pregnancy and first start looking at, how regulatory T cells change throughout pregnancy, right? Moms don't have frequent lab draws necessarily, but there have been some studies that um, look at circulating levels of Tregs, but they usually don't start until the mom comes in with contractions or she's noted to have dilation on one of her exams. So I think starting earlier in pregnancy, understanding better how that changes. So is it that these moms who are at risk for preterm birth that they have less of an expansion of their Tregs from the get-go? And that just kind of low level is maintained like that? Or is it they have the normal expansion and then it just starts to tail off a little bit earlier? And so it leaves them more vulnerable to preterm labor and delivery because it kind of reaches a critical threshold earlier in gestation than other people. And I don't think that we have a great answer for those yet, but there are a lot of people doing amazing work looking at some of these same questions. And I would like to be a part of that. And I think it is important work.
You also mentioned just the function, like if they're even if the number is normal, but they're not functioning normally, are there good assays for just testing regulatory T cell function, or do you just look like basically the immunotolerance is lost because they're not functioning, but the number is normal? How do you evaluate that?
So most people, so Nardi Gomez Lopez has published some of this work, and basically what they do is they isolate effector T cells, and they take the Tregs from the decidua in general, and then they kind of do dilutions, right? So you have fewer and fewer Tregs and see how effectively they can suppress the T effector cells. And so it's almost like antibody titrations. They see what's the lowest effective dose, and then that gives them information for how effective the Tregs are at inhibiting that response. And so that's how the work's been done so far.
Do you think that much of the effect of the cell of the Treg is in a canonical sense, meaning they're suppressing the inflammatory effect of other cells? Like this, how I would typically think of a Treg secreting cytokines or potentially directly interacting with other cells to suppress their inflammatory activity? Or do you think there may be a more unusual role for the cell in the placenta and uterus? And I'm thinking of a colleague of mine who studies NK cells in the uterus and placenta, and they have the NK cell has a very unique function just in that environment that it doesn't have in more canonical senses. So does the Treg have this kind of are there any unique activities of the Treg in pregnancy?
So that's a great question. Thank you for giving me one more thing to now think about as
Yeah.
I'm doing these experiments. I haven't really come across a description of Tregs of, a very specific pregnancy related. The thing is, it's possible though, right? Tregs respond to progesterone and hormonal kind of cues. And they also, they act as this, not only do they secrete their own cytokines, but they act as a sink for like IL-2 and things like that. So they inhibit growth factors to other cell lines, right? In the context. And the environment in the placenta, we have to assume it's very different from how things exist in peripheral blood or things like that. So I wouldn't be surprised, but that's not something that I know is well described at this point.
Yeah, fascinating. It feels like a whole unique world, the placenta especially, but also the uterus. Um,
Well,
so
that's why a lot of
yeah.
people say that they want nothing to do with the placenta.
Is that right? Because it messes up all the other experiments or
Because
something.
everything's just different. It has its own
Yeah,
rules.
I think that's why
yeah,
it's so interesting. Obviously, it's so like necessary for our survival. But yeah, it is complicated. It seems like there's so many systems that have to be imbalanced to for it to work normally. And if they're just subtly perturbed, things can go really wrong. It is one of those examples of like it's just amazing that it ever works ever, you know? Um, just because we are developmental biologists, or at least I am, and I always think like the way you study the function of something is to like knock it out, and so you're reducing the function of the T regulatory cells, and that helps in one way. Can you what happens if you like turn them up? Like if you have too many of them, or if you turn up their function too much, like how does that affect how things work? Or do you know anything about that, or is that an issue?
So, great question. I don't know that we have like a functionally relevant model in human disease, or I'm not even sure if there's a great mouse model of that.
It's totally outside my world. I don't know. I was just wondering, like, I mean, that's just how we think about, stuff and, like, how pathways get activated, like, okay, if you remove the pathway, we can see what happens and learn about it, and then if you overactivate it or activate it in a context it's not normally in, what happens? I was just wondering, especially just thinking about, like, if to test Ben's question, like, is it a canonical issue? Like, so if the issue is just maintaining a balance of immunoregulation, and if you turn it up too far, like, then the, I guess, animal gets susceptible to infection or things that you would wanna be able to have some kind of an inflammatory reaction to.
Right. And that's like, part of the perfect balance that pregnancy needs to get to, right? You need to have enough
Yeah.
Tregs to dampen down the immune system enough to allow that baby to grow and flourish, but not enough that the mom's so immunocompromised that you're putting the whole pregnancy and the mom at risk. So it's a good point.
Thank you, Jackie. That was really excellent. You're an amazing teacher. Like I'm not an immunologist, but way less than either of you. And I feel like I learned a lot and could easily understand how this is so applicable for such a common problem. Like preterm birth is the most common reason why babies that we take care of in the NICU are admitted. And it's really interesting to think about the many ways that, preterm birth can happen and, how we can learn from our patients and then design experiments to study the things that we encounter all the time. I was just wondering before we finish today, just because you mentioned the PSDP is a program that was really helpful for you getting established in your career. Could you talk maybe just a little bit more about that or other programs that you have like been a part of that you thought were really helpful for helping you develop your physician scientist career? I think that's another thing we're just really interested in is how do we enable more people to pursue this career path and just learning about experiences from people like you is really helpful.
Yeah. So the PSDP was a program that's kind of built by AMSPDEC and supported by the NIH. And I know that there is a lot of uncertainty about how that works right now, but what it is you either apply in your last year of residency for most people, or your first year of fellowship if you are like in critical care. So, like, I applied my first year of fellowship because I was a neonatology fellow, and it basically allows for 2 years of protected time of research. And so not only did I have that protected time, I also got to meet with other, pediatric scientists at the similar stage in their careers who had a goal of running their own lab. And so it really built that community. And like we still have a WhatsApp like group chat where we will update like, I just got one the other day. One of the guys his wife just had another baby. And so, like, we still keep in touch. And, whether we're talking about, like, oh, I just put in my K08 or my K23, or, oh, I'm, moving institutions and setting up my lab, hiring my first technician. Um, I think that community, at least for me, has been a huge thing to be able to be like, well, the path is hard and there are times where, it gets a little rough, but you have a group of people that you can reach out to, um, and kind of experience that together. Um, and so the PSCP was wonderful for the research aspect and the giving the community. And I think that that's probably the biggest impact it's had for me. Um, and then there have been other programs that are incredibly helpful. So, like as faculty, I was on a K-12 here at Indiana, which having the support of your, chair as well as division chief is I don't think you can do research without that support. And so that's been instrumental. And then there are other smaller things like the Perinatal Research Society. They do like a grant writing course. And so I did that, I think my first year as faculty, and like worked on my KO8 submission, met a bunch of great people who are also interested in similar stage of your career. Um, and got feedback on that from people outside of my institution. So I think for me, the building the community and networking through opportunities like that has really probably given me the most staying power in this career that at times it can be quite uncertain.
How just how I mean, one thing is just how did you get introduced to those, different programs? So, like, the PSDP, had you heard about it before? How did you get introduced to that?
So the PSDP, so people that I went to and did my MD PhD with, who were a couple years ahead of me and went into pediatrics. So Simon Rhodes, who's a peds hemonc doctor, he was a couple years ahead of me, and it's something that he did. And from the get go, he's like, "This is a great program." Wade Clapp, who's my chair, is also was really big on kind of supporting the PSDP, and so it was kind of something between those two influences I knew was going to be on my trajectory to apply for. So it's, I mean, a lot of it's about the people, right? Networking and having those people that are going to look out for you in the sense of being like, hey, keep this on your radar, Jackie. This is going to be important for you. And, a lot of times they're right.
And PRS, the Perinatal Research Society, that's a perfect environment for the work that you do, especially, but I love the, like, overlapping and cycling leadership of it. So there's, like, a neonatology group, there's a maternal fetal medicine group, and then there's sort of like a basic research group, and they sort of cycle through those 3 things in terms of, like, leadership of the society, but also, like, the focus of the meeting. But that grant writing course, like you said, I thought that was outstanding just to be able to take I don't know if you did this, but I took an Ames page there and then sat with someone, I think it was Kurt Albertine, who read my Ames page and then gave me feedback. I went to another room, edited it, brought it back to him again and again and again. And that, it really felt like I made a lot of progress. And then also you get a lot of other advice and another chance to network, just like you said, with people who are at a similar stage.
Uh, I had a very similar experience. I love that medium. I'm actually going back this year, and I'm going to be
That's
presenting
great.
my work as like one of their little short sessions. So I'm excited about that. But it's a small enough meeting that you can actually talk to everyone.
Yes.
And I really appreciated that, especially as someone who was kind to break into kind of the reproductive immunology space, right? That was a newer aspect to as I started to establish my lab as a faculty member. So it's great to meet key people in that setting and then get just great advice on grant writing in general, right?
Well, I wanted to also ask about Indiana
Yeah.
because I've only done a few interviews so far, but two of them have been with up and coming successful physician scientists at Indiana, you and Kristen Noble.
I was gonna say Kristen Noble.
And and I I just have to think that there's something special going on there. And I'm curious what, if that's true, and if it is, what you think might be the keys to success in your environment at Indiana.
I very much think that's true. Riley is a very special place, and one of the people that I've identified as helping to make it that way is Wade Clapp. So he was the director when I came on, and he's been the chair of peds for forever. He's just stepped down this year and passed the torch. But between him and Laura Hainline, who's our division chief of neonatology, who also has a background in being a physician scientist, I think the way that they buy in and support people is incredible. So, like, Wade was on my scholarship oversight committee as a fellow, and I can remember after presenting, like, Wade came up to me afterwards and he's like, "Ah, Jackie, you're doing really cool stuff. I'm very excited for you." And he's like, "Yeah, I've got your sign-on package like all ready to go for when you're faculty." He's like, "I want you to interview everywhere, but I'm going to make you the best offer and I'm going to keep you here at Indiana." He was right.
That's amazing. Yeah.
Great to have your chair in your corner like that early on. That's awesome.
Doesn't get much better than that. But
No.
really, the way that the support you don't get everywhere, right? And that investment in you as an individual and in your science is pretty special.
Yeah, it seems like it's really working well. I mean, it seems like there's been a good program that has like helped you develop the skills that you wanted to be able to develop to establish a really exciting research program addressing a very common problem that we don't understand very well and use techniques that I think will really be able to address the underlying mechanisms and help us learn and make progress. So super exciting. Oftentimes to finish, we'll ask just like something that tries to demonstrate our humanity and indicate that we don't spend all of our time working in the lab. Um, Jackie, is there anything that you like to do either personally or like as a research group or maybe with your family that is just something great to do outside of work?
Well, with my family, we like to go rock climbing. A lot of
Oh,
times it's
wow.
indoors, but sometimes I haven't quite taken my girls out to Red River Gorge, but I've been climbing there myself. So enjoy that. And as a research kind of community, my lab's really small right now, so it's me and a technician, but we exist in kind of the Kaplan lab sphere. And so we'll do the things like going out to meals every once in a while. And if we're at conferences together, or actually, um, a quick anecdote. So the uh, pediatrics, so PAS, which is the big pediatric research conference, is usually in the same city as AAI, which is the immunology research conference, but they're usually like 2 weeks apart, like close enough and in the same city that I never go to both of them in the same year. But so last year I went to PAS and it was going to be in the same convention center as AAI. And so I took a little post-it note and I wrote the lab a little note and I stuck it on the wall in the conference center and took a picture of it and just to see if it would still be there for them when they were there a couple weeks later. And they went on a little scavenger hunt and found it and took a picture of it and texted it back to me. So that's just a little thing that kind of shows that the community that we try to build and the connection that can be so important in a research lab environment.
Yeah, I totally agree that community is the essential thing. Like having a common bond and shared experience, I think is so valuable. It makes the work much more interesting and exciting to be able to do.
Exactly.
Well, Jackie, thank you so much for taking time to talk with us today. It was so great to learn about your work and, the motivations that you've had. And just, I mean, you're a great role model. So it's awesome to hear how you've gotten to where you are. Super excited to hear what you're doing next. And one thing we didn't get to talk about was the Midwest SPR meeting, which is coming up in just a couple of weeks. But I know you're involved in hosting that, which will be great, and the career development session, which I'm sure you'll have a great influence on the people who are coming there to present. But thank you so much for taking time to talk with us today.
Thanks for having me. I really appreciate it. And I hope to see everyone in Indiana for the Midwest SPR.
Awesome. Thanks, Ben.
Awesome. Thanks, Jackie. Yeah, thanks,
Thanks,
David.
guys. Appreciate it.
Bye.